Hi, this is Camilla Teng, reporting from the lab of Jeff Bush at University of California, San Francisco. In this episode of video summaries of amazing work featured in the Journal of Cell Biology, we are talking about fusion, tissue fusion. In this current study, we've developed a new playground for understanding cell behaviors of morphogenesis. Think 3D. Think complex topology. Think lips. That's right. The lips that we use to speak and smile every day underwent a complex process of development to become what they are. We know from static imaging of mouse embryos that at one point of development, they look like this, with swellings called the medial and lateral nasal processes that surround the future nasal cavity. These processes must then come together to make a continuous structure that is our upper lip. And if that doesn't happen properly, we get cleft lip, a common birth defect worldwide. To uncover the cellular mechanisms that drive this fusion, let's join our correspondent in the field and see.
Thanks, Camilla. Using our live imaging setup, we are able to survey actin dynamics in craniofacial tissue fusion in real time. We see dramatic actin enrichment at the fusion site and actin cables pulling the tissue together. Back to you, Camilla.
Hand in hand with actomyosin contractility is cell adhesion. And indeed, P120-catenin, a core adhesion junction protein, turns out to be very important in the genetics of cleft lip in humans. There have been insightful studies picking apart the functions of P120-catenin in cells, but less in mammalian embryos. We adapted an electroporation-based gene editing approach to generate an allelic series that surgically interrogates the relevance of different P120 functions in lip fusion. Turns out, abolishing P120-catenin and cadherin interactions in the epithelium will result in cleft lip, similar to what happens to complete deletion of P120-catenin in the epithelium. But riddle me this, how is it that losing P120-catenin or P120-catenin cadherin interactions in the epithelium would disrupt fusion, but removal of E-cadherin from the epithelium has no effect? The answer is P-cadherin. By decreasing combined alleles of ECAD and PCAD, effectively titrating cadherin levels, we find a critical cadherin threshold is necessary to enable fusion. With our collaborators in the Leslie Clarkson lab at Emory, we have also found evidence correlating mutations in PCAD with human cleft lip. Our working model is that the localized enrichment of actin observed at the fusion site produces forces necessary to drive fusion. And this tension may cause tissue sensitivity to reduce cadherin-mediated cell adhesion. In other words, we think that the fusion site acts like a fault zone. The epithelium is actually fine with some reduction of cadherin cell adhesion, except when it is challenged by actomyosin tension. This concludes our video summary. Thanks for watching, and please remember to check out our paper for more details.